Welcome everybody to this session about childhood cancer biobanks. Actually, when I attended the first meeting a couple of years ago, 4 years ago, then it was one of the main topics, and I must say I'm deeply impressed how things have evolved during the time, so especially in Latvia. It's really wonderful. And our first speaker is Johanna Sandgren. She is the director of the Swedish Childhood Tumor Biobank in Karolinska in Sweden. And actually, when we started our Norwegian childhood cancer biobank, that was our first place to visit. So we went to Sweden, we learned a lot from your biobank, and it was really a huge help to get started and to get established. So, and I see you do a wonderful job there, so I'm looking very much forward to your talk. Thank you.
Is it on? No? Yes? It's on. Okay, thank you very much. Thank you, Lars, for the introduction and the possibility to speak to you. As said, I will present the Swedish Childhood Tumor Biobank, a natural tissue omics research resource for pediatric cancer. And I probably need this to change slide. Yes.
I think it's the other way around.
Other way around.
Yes, okay. So a brief background about pediatric and maybe some about the situation in Sweden. So in Sweden we have around 10 million inhabitants, and every year around 350 children are diagnosed with some kind of cancer. And today more than 85% will survive the disease, but it's still the major cause of disease-related deaths among children. And for several of the diagnoses, it has been reaching the survivor plateau, because if you look at, how do I then point? This, okay, thank you. So these are some survivor plots from the Swedish Childhood Cancer Registry, with survival for different cancer diagnoses already from the 50s. And up to 2015, actually. So, of course, with the introduction of chemotherapy, the survival increased a lot for all kinds of cancer. But what I mean with the survival plateau is that you see for several diagnoses, the curve has flattened out. And also importantly, for most of the survivors, 70%, they suffer from long-term side effects from this treatment. And moreover, I want to point out, as we have heard in several talks in this meeting, that childhood tumors differ from adult ones. They have a different biological origin, so we have different kinds of diagnoses in childhood tumor compared to adults, and of course they occur during development. And also importantly, they have much less and also other genetic aberrations compared to adult types. And there seems to be in general less interaction with the immune response. And also recent studies have shown that maybe a little higher fraction of the childhood cancer cases are due to predisposition if you compare to the adult ones. Still, it's rare, maybe 10–15%. So this is, of course, then arguments, and as we all know, that more research are needed for these malignancies. So that's also the background to why the Childhood Tumor Biobank exists. Because here is the aims and purposes of the biobank, and it's called Barntumorbanken in Sweden, we usually say BTB. So the aims are then that we are a research, or in short what the biobank is a research and healthcare oriented biobank infrastructure. We are a national, in Sweden, done sample collection for solid pediatric tumors. There is a sample collection in Uppsala collecting all the leukemias, so we take care of 2/3 of the cancers in our biobank. And what we also do, and has been doing for almost 10 years, is to characterize the samples in the biobank genomically. I will tell more about that. And the main purpose with this infrastructure and sample collection is that we can distribute these collected samples and also the data to authorized researchers to facilitate research. And we are also fully funded by the Swedish Childhood Cancer Fund. So the overall aim is, as I said, to support research and of course for the benefit of the patients. So a bit more in detail about the sample collection and the analysis. So if we start over here, so as a biobank and as a research sample collection, you need ethical permit and patient consent to inform the patient and the parents, since these are our children. And of course, biobank samples, they should be traceable and they can only be used for specific purposes. So we have, of course, this set up. And we collaborate with the 6 Swedish childhood cancer centers and the different divisions in every centra mentioned here. And the genomic characterization that we do includes whole genome sequencing, and that is done in collaboration with SciLife Lab, which is a big life science infrastructure in Sweden. And of course, since cancer is due to genetic changes, this is an analysis that can give a lot of extensive information about the tumor and answer a lot of relevant questions, like the origin, mechanism, progression, treatment response, and find new targets, refine diagnostics, predisposition risk, and of course, often in combination with other analysis. So this genomic analysis generates a lot of big data. So we have also done developed an IT infrastructure to store the data, analyze it, and also be able to share it. Another picture summarizing some of the work in the biobank. So when the samples are then sent to us from all of these 6 centras, and then what we do is that we register them in the LIMS system, we can prepare and we store the samples. And then, as I said, we do the genomic analysis, whole genome sequencing. We also do RNA sequencing on all the tumors. Whole genome sequencing we do on the tumor and blood. And we also do methylation arrays on the CNS tumors and sarcomas. And these data are then bioinformatically analyzed with different pipelines, also in collaboration with SciLife Lab. We have databases developed for storing the data and metadata. We also collaborate with researchers with the data and also help out some clinical national studies in Sweden. And we have these formal processes then also to be able to give researchers access to the data and samples. And here is listed also more different sample types also from the different clinics. And what is new also in recent years is that we don't only collect samples and information from the patient about the diagnosis and so on. We also collect genomic data now, because if you have listened before today to Gustav Ljungman about the Genomic Medicine Sweden project, this analysis, all of these are now done in the clinic in Sweden. So we can collect this data to the biobank. So this is the third point here, that we can collect this data now from the healthcare, which is of course very good for the immediate care for the patient. So what about what do we have in the biobank? So we have been collecting for a bit more than 10 years. In this graph, you can see the cases collected every year, the number of individual patients, and the blue part of the bars are the brain tumors, and then we started a few years later with the other solid diagnosis. And the line here is representing from the cases where we have both a tumor sample and a blood sample. It's often easier to get the blood samples, but sometimes we cannot get the tumor because it's needed for the diagnosis. We only collect the surplus material. And I want to say it's a very good collaboration with all the centers. We get consent and biobank samples from more than 90% of all the children in Sweden with cancer. And the status recently, so this is per centra, so in total we have now more than 2,300 unique cases in the biobank. So that was about cases, and if we look number of samples that we have registered in a biobank, because we collect different sample types from the same patient. So I have some numbers here. So if we look in our LIMS system, we have more than 32,000 samples, and in addition to that, extracted DNA and RNA, additional 22,000 aliquotes. We have also some protein fractions and viable cells in cell suspensions. So here I've also listed a different sample type. We have focused on the fresh frozen material since we are doing the genomic analysis, but we also collect paraffin embedded and blood, and we collect also from the parents, and then tumor cells and CSF. And of course, it's important also to collect metadata connected to the sample. So I listed some variables here, information from the patients, and of course, diagnosis and the QC metrics. So we are a population-based biobank in Sweden for CNS tumors, and these pie charts are then showing the distribution and the frequency of the different diagnoses in the biobank. So of course, the more common diagnosis, we have more samples, so it's CNS and solid. I want to say a few words about the genomic analysis program. So we do this Illumina sequencing, the short-read sequencing, since 2015 in collaboration with SciLife Lab. We process and store the data, and we have also other collaborations with platforms in Sweden to store this sensitive data. We analyze it, and we can then distribute these data sets for researchers. But what has happened then in the recent year, as said, this Genomic Medicine Sweden project has started. So now the sequencing protocol is now done in the clinic with different facilities called clinical genomics. And their data is then in clinical reports to the pediatric oncologist. And this project has been going on as a study since 2021. And now more than 1,100 patients are included in this. And all this data, as I said, that is generated here also goes into the biobank and can be available for further research. And as Gustav mentioned this morning, it's no longer a study to report the variants, it's now implemented in the clinic. The generated data that we have now, focusing on this genomic data. So we have sequenced more than 800 cases where we have whole genome sequencing data and RNA data. And now for this GMS study, we have more than 600 cases at the biobank, but we have not, because as I said, there were more than 1,000 patients included, but there is a delay for us to receive the data in the biobank. So as I said, this is a big data. So now we have, I guess one copy of the data is 2.6 petabyte. So that's why we need this advanced IT infrastructure for, of course, also backup of the data. And here also we collect the important metadata for all the genomic data. Next slide is about research applications to us for samples and data. And since this is human sample, sensitive data, there is a formal process. Every researcher needs to have an ethical permit relevant for the application. We have a medical legal review for every application that you're authorized to use this data in research. And we also have a scientific evaluation, so we are sure that samples and data are used in projects that actually will be in the end hopefully some benefit for the patient, some quality assurance. And of course we check if we have the sample and data for that request. So the list here are we started with this some years ago, maybe 5 years ago, accepting applications because we wanted to build up the biobank with samples and data. And now it's just growing all the time. But in total now we have 22 different research projects that has been approved for samples and or data. Actually most for data, which is good because data you can copy endlessly. Samples are more precious. So there are a wide variety of, this is mostly Swedish projects. We have one request from Heidelberg for data, a low-grade glioma study, but the most Swedish projects. Different application using data and samples, single cell studies, xenografts. We have this, also this project, if you can see it says digital pathology, because we also collect the digital slides of the tumor section, so they are applying AI on these images for improved diagnostics. We have pharmacogenomic studies, and we have also studies focusing on the predisposition to cancer due to the TP53 mutations, and then different projects focusing on different diagnoses like sarcomas, neuroblastoma, lymphomas. So. Also, these are a list of clinical studies that we can help with different biobank services also listed here. One that you recognize might be the INFORM study. So some slides on ongoing developments and pilots. So what we are planning now is to introduce new sampling protocols. And that also requires ethical permit updates and new consents and new biobank agreements. But we will start soon with blood plasma, because now we collect the whole blood and extract DNA. We would like to have the plasma collection with also several time points, and also extend the collection of CSF and viable tumor cells. And of course, this is to allow for more studies, like different biomarker studies and single cell analysis and different models and functional assays and so on. And we're also planning some pilot projects internally on the material, proteomics on tumor samples using mass spectrometry, because this will add an additional data layer, integrate with the genomic and transcriptomic data that we have. And of course, proteomics can be important for a lot of different questions. And we are also now starting long-read sequencing pilot projects in the biobank with the Oxford Nanopore technology, because with the long read we can probably find more confident detection of structural variants. And also, as you might know, in parallel you get the DNA methylation status, which is very useful. And then I said data portal and databases. Some internal work that we do, because it's important for us as a biobank to be very well structured, also with metadata, not only the samples. So this is a flow for the data that we are constructing in this data portal. We retrieve information from the LIMS and from Excel files to process this into this user-friendly interface. And the aim with that is that the data portal can bring the data together. As I mentioned a few times, there's a lot of metadata to the cases and to the analysis that we do, preparation of the samples, and of course also to the genomic data. So we have a prototype for this, the web interface, where we also can visualize the data, and we will also add information of what kind of projects data has been used for and the samples. And moreover, we have constructed variant databases for the somatic mutations and for the germline mutations. And this is just a screenshot how it can look for looking for TP53 mutations in germline and for somatic NF1 mutations. So one of the last slides then, so in summary, we want to stress that biobanks really can enable research and of course also precision medicine, which many of the projects that we have approved samples and data for are working for. And we also, of course, wanted to continue to develop the biobank so we can have an increased use of the samples and data. Because, of course, we think we are a valuable population-based genomic biobank. And we are a dedicated group working with this at the Karolinska Institute, mentioned here. And, of course, we collaborate, importantly, with all the 6 centra and also locally with pathologists, and we have a steering group and other collaborators. And of course, we would like to thank the patients and the parents for participating.
Thank you, Johanna. I do have a question for you, but we are having this question and answer session at the end of the session, so I will leave it for later. And now I would like to kindly invite our next speaker, Professor Torben Mikkelsen, Associate Professor, Department of Pediatric Oncology at Aarhus University Hospital, Denmark. The topic of his presentation is Building a Biobank for Childhood Brain Tumor Research and Collaborating by Sharing Knowledge. So please, Professor, the floor is yours.
Thanks for inviting me, and thanks for the kind introduction. So I'm actually not a professor. but just an associate professor. And I like to think of myself as just a common clinician who wants to help progressing this field by collecting data. So when I give this talk, then please think of yourself as a fellow who can do the same as I can with these samples. So I'm from a small center in Denmark. We have 2 centers where we treat children with brain tumors, and I'm from Aarhus. I'm going to tell you about how we started a national biobank for children with brain tumors, and the first part of the talk is about the practical aspects of how you collect the samples, especially the spinal fluid, and the second part of the talk is what you can use the samples for. Because I think it's important when you have a biobank, then you need to do some research experiments with the samples in order to find out if you have collected the samples in the right way. So when you think of a bank as a place where you store your gold and you keep it secure there, whereas a biobank is also a place where you store your gold, but it only has value if you're able to get the gold out again, use it for research, and especially use it for collaboration as well. So in Denmark, we have about 60 new cases per year with brain tumors. And if we collect all these patients, then not even in 100 years from now, we'll be able to do a study on the rare brain tumors. So if you can't collaborate with others, then your biobank will have limited value, I think. So the first thing you have to make sure is that the patients, they give consent for collaborating and sharing the tissue when they contribute to your biobank. Also, an important thing is that you need to have a standard operating procedure for how to collect the samples, because if you're a country collecting samples from many different hospitals, then you probably have different ways of doing this, and even in your own hospital, the staff is changing over time. So make sure that when you start and when you develop and progress, then you have a very good and detailed standard operating procedure for how to collect the samples. Last month we had a meeting in the Nordic brain tumor group where we decided to try and harmonize the way we collect the samples and the way we process the samples in the lab, so we'll be able to collaborate across borders, maybe not by sharing the samples, but at least by sharing the data from samples that have been collected the same way. So during the 4 years we have collected data, we have gotten consent from almost all patients. We have sampled blood from all patients before surgery, and at some point during treatment we have been able to collect spinal fluid in 80% of the patients. 70% of these patients had spinal fluid collected before surgery. And this is an important thing, because if you collect spinal fluid after surgery, some of the valuable biomarkers might already be gone after a couple of days. I'll come back to that. Then we collect tumor tissue as well. If it's a big tumor, then the tumor tissue goes directly from the tumor to our molecular lab. If it's a smaller tumor where the pathologists have to prioritize, they will first take tumor tissue for diagnosis and then later on for our biobank. But for almost all patients, we managed to get the tumor tissue for whole genome sequencing. Then an important thing when you do a biobank on pediatric brain tumors, that is to do methylation analysis. Because as Stefan Fissler told us this morning, the pathologic evaluation of the tumors is not always enough to get the right diagnosis. So you don't want a biobank where you don't really know what you have, which kind of diagnosis you have. Then you'll never be able to pull out the data and combine the results. Then the most important thing when you start a biobank is to have a dedicated research nurse. When we started our collection of spinal fluid and the biobank, I found out that all patients with brain tumors, they of course come in the afternoon because they go to the physician because of focal neurologic symptoms, headache or vomiting, then they get an acute or subacute MRI or CT scan, and then they rush to the hospital with the diagnosis of a brain tumor. And many of these patients that get operated or have surgical procedures as third ventriculostomy the day after they arrive, So if you want to collect the spinal fluid on all patients, then you really need to have a dedicated nurse who can be aware when a new patient comes into the clinic and then follow this patient to the OR, because the surgeons, they'll be focused on something else. So they'll forget to sample spinal fluid for you if you don't have a nurse standing next to them yelling, "Give me spinal fluid, give me spinal fluid." And she needs to come there with the blood tubes also, because otherwise you won't get the right tubes and you won't get them handled the right way when you take them to the lab. Also, when you do a lumbar puncture, day 10, 13 after surgery to find out if the patient, they have metastatic disease, then many times it's a younger physician at the department who will do this lumbar puncture, and this physician might not be aware of the biobank. So you won't get samples for your biobank unless you have a nurse who's there. So spinal fluid and circulating tumor DNA in particular seems to be the new gold for childhood brain tumors. These small fragments of tumor DNA is released from apoptotic tumor cells and also from other apoptotic cells in the body. They have a really fast half-life, so if you don't sample before surgery, then you won't have any circulating tumor DNA. And also if you sample after surgery, you'll have a lot of noise in your sample because there'll be tumor DNA. There'll be free tumor DNA from all the normal cells as well. We always take 10 milliliters of spinal fluid when we do a lumbar tap or when we extract spinal fluid by an Ommaya reservoir. And then we divide it for clinical examination of tumor cells and then for research purposes. When we have a patient with an external drain, then I sample all the time, at least every day, 20 milliliters. Because then my idea is that then we will have a biobank one day with a lot of spinal fluid that we can share to other projects, and then maybe we can do some of our projects, some of our own projects, but we can also participate with spinal fluid to other people. And typically the patients come with high intracranial pressure, then they'll get the external drain, and then we'll plan surgery with the right team within 2 or 3 days after that, and then we have 2 or 3 days to sample all the spinal fluid that we can get. We don't really know if location matters as if where you collect the spinal fluid, but we have a SIOP study where we want to collect paired samples on 30 patients and then see if the level of ctDNA differs depending on where you sample these spinal fluids. So the most important thing for the level of ctDNA is probably the tumor type and disease burden. In this diagram here, you can see that we have a lot of patients with almost no circulating tumor DNA or cell-free DNA. They're on the bottom there. And then it really doesn't matter if you sample 2 milliliters or 6 milliliters or even 10 milliliters, you still don't have enough cell-free DNA for your analysis. But what you see is that many of the patients actually have quite high levels of tumor DNA, and these are the malignant tumors. And you will have enough to do all these different analyses, as I've shown out here in the side. If you collect the spinal fluid in a sterile tube, then you probably need to keep it on ice and get it processed right away, because the half-life of circulating tumor DNA is so short. The advantage of collecting the spinal fluid in the sterile tube is that then you can also collect CSF for proteins and RNA and circulating tumor cells, whereas if you use the preservation tubes, then you'll be able to get the ctDNA out of the glass, but you won't get the other things. I recently looked at our data and found out that actually almost half of the patients, they had their spinal fluid collected in the preservation tubes because the samples were taken after 3 o'clock or in the weekends or in the evenings. So you want to be able to handle both. And if you want a sample shipped from one of your collaborative hospitals, then you can use the preservation tubes. So the preservation tubes have not been evaluated on spinal fluid, therefore for blood samples, but we have a SciAp working group called Pre-Analytic Working Group where we want to evaluate this and come up with an evidence-based guideline on how to sample CSF and also eventually which method would be the best for which disease. The most sensitive method for detecting circulating tumor DNA is digital droplet PCR. With this method, you capture every small DNA fragment in a droplet, and then you sequence the droplets with fluorescent probes. And in this assay, you can see that the BRAF mutation then gives you a blue fluorescent signal, and the wild type gives you a green signal. The lab report will look like this. You have a lot of blue dots, which are positive for the tumor-specific mutation. The green dots are the wild type allele, and then you have the gray dots with that droplets without any tumor DNA. And in this patient with a malignant brain tumor, you can see that she had 28% of her droplets were positive for the tumor-specific DNA. And then theoretically, you would be able to follow this patient with digital droplet PCR over the treatment period to see if the tumor relapses and if the treatment you give her has the effect that you expect. We also have this method. I think everybody today has shown something with nanopore sequencing. So we have this project where we try to get a really fast diagnosis pre-surgery by using the ctDNA. We use the nanopore, which pull the DNA through these nanopores, and as you see here, within 5 minutes we had DNA methylation data on more than 2,500 methylation sites, and the diagnostic calibration score here was 70% already after 5 minutes, and this was for medulloblastoma. The nanopore sequencing seems to be very good for medulloblastomas and less robust for some of the other tumors. So just a few slides about blood samples. We know that ctDNA from brain tumors also enters the blood if you have a large tumor that are malignant. But most tumors, then you won't be able to find the ctDNA in the blood from these patients. If you have a patient with a large supratentorial tumor where you can sample CSF, then maybe it's a good idea to draw blood. And if you take 20 to 40 milliliters of blood, then you'll probably be able to extract enough ctDNA for your research purposes. The embryonal tumors with multilayered rosettes is a very rare tumor which we actually haven't seen for many years now. But in this tumor, it's been established that the microRNAs are important and good to measure the tumor, the disease burden, and the efficacy of the treatment. And then a small comment is that yesterday the Nobel Prize in Medicine was awarded to the scientist who discovered the mRNAs and the value of this. So maybe this will boost this area, and we'll know much more about MRI in a few years from now. We have a small project where we study the extracellular vesicles in the blood. So these are membrane-bound vesicles that are continuously secreted by the brain tumors, and the half-life is really fast. So if you can capture these vesicles, then you will have a measure of the tumor stages right now. And you can measure the proteins and the metabolome and the RNA in these vesicles. You need to sample them or to handle the plasma through ultracentrifugation. So it's a bit more specialized than just looking at the ctDNA. We have a small study where we showed that Syndecan-1 in these extracellular vesicles is increased in the large tumors and in tumors and the malignant tumors. Last slide. Don't forget to collect the buffer code. There's so much interesting research on the diversity of T cells and how the peripheral immune system actually influences the tumor and the response to treatment. Although we know that most pediatric tumors are cold tumors and the immune system don't actually enter the tumor cells. It looks like the diversity of the T cell receptor is a good prognostic factor for many adult cancers. So collaborate, collaborate, collaborate. Remember, you need a research nurse if you really want to collect samples on all the patients, and the best patients always come in the weekend or the holidays, right? So you especially want to get those. And then get started with your research, because then you'll know if you sample the correct way, if you sample the right things that you really want to do research in, and then it will give you an insight into how you can collaborate with others. Yeah, blood has potential. Write to me if you want to collaborate or have some of my material.
I actually will use that opportunity, definitely. Thank you. It was very inspiring, and liquid biopsies are a hot topic, so we missed the applause. Yeah, sorry. So I would like to kindly invite our third speaker, which is Daniel Naumovas, Head of Biobank, Senior Researcher, Vilnius University Hospital, Santaros Klinikos, Lithuania. The topic of his presentation is Biobanks and Personalized Medicine, Unlocking the Future of Tailored Healthcare.
Yes, hello everybody. Yes, I will speak about biobanks and personalized medicine, as you know. So first of all, just a short introduction. All of us know what is personalized medicine, but we have still here some patient representatives. I will just shortly mention. So imagine we have a 40 size shoe, and if we will give it to the person who has 40 size, it obviously works. If we will give it to the person who has a smaller shoe size, it will work, but not as comfy as the direct one. And obviously, if we will give it to the person who has bigger foot, it will be not as comfortable. So the same thing with the drugs. For some they can work, for some they cannot work, but they won't give many side effects, and for some they will not work and they will give side effects. So right now, why does treatment work for some and not for others? We need to understand that one disease is not equal to another. The same one cancer is not equal to another cancer, and usually people forgetting that. So how can we find the difference? Obviously, biomarkers and different omics. So this is a basic thing. One of the starting material is DNA, but we should not only focus on DNA, but also RNA, proteins, and other omics. And for that all, we need samples and data. So you know where I am leading. It's obviously about biobanks. But before that, there is one interesting research. So really interesting in cancer biology publication, they analyzed around 200 experiments and they tried to reproduce those studies. And only a quarter of those experiments were able to be reproduced. And main reasons why there was a poor reproducibility, so poor description of the methodology, publication bias, sample size, sample quality, and design quality. So, and usually people in this kind of papers write that samples will be collected by the standard protocol. And I love one quote from the Adams family, Morticia Adams, the mother said, what is standard? So what is standard for spider is a chaos for fly. So we shouldn't use this kind of wording. So you all know what's biobank, and usually when I'm speaking about biobank, I'm showing the library. As the librarian knows where which book is, the biobankier knows where which sample is and what data it has with itself. So nobody showed it, but usually biobankier is showing this. So it's not a new thing. It's a new thing maybe for Latvia and Lithuania, relatively new thing. But overall in the world, biobanks been for a long time. And even Times Magazine wrote that one of the 10 ideas changing the world right now is biobanking. And it was in 2009, so already 15 years ago. So there's a small example about our biobank. So the patient comes to hospital, obviously he's not coming for science, he's coming for treatment and getting better health. So whenever a patient coming, he can sign a consent and agree to participate in biobanking activity. Right now in our biobank we have more than 10,000 people consented. Then the medical procedure is happening, and usually all biobankiers should know in what hospital they are. For example, we have 35 departments, so we know that we can collect all kinds of diseases. If you are smaller hospital, you should focus on one kind of disease at the beginning. Then the sample collection happens, and biobanks can collect everything from blood, then derivatives like DNA, RNA, nasopharyngeal samples, saliva, hair, nail, everything is collected for different purposes, for different collections, different samples. And there also should be always biobanking on demand. When a new study appears or new researchers come to you and want to do research with you, you should definitely think about storing samples or storing new samples or in a new way. Biobanks about standardization. So biobanks should use standardized protocols. So we are aiming for the obviously BBMRI, ERIC quality management system, and also 2 ISOs, one of the documentation and another biobanking, and all biobanks should also focus on that. Samples should be stored at ultra-low temperature. I would suggest focusing right now, the lower temperature the better, obviously. -80 should be used for the molecular diagnostics, like DNA, RNA, proteins, lipids, but if you would like to work with viable cells, you should obviously focus -150, or liquid nitrogen temperature -196. So right now we have collected around more than 200,000 samples. And then samples should be distributed. Obviously, collecting samples is not the aim of the biobank, but to give away those samples. And then obviously scientific results are coming. And the best thing about biobanking that usually people are forgetting that, for example, when we are storing from one blood like 5 aliquots, right? Then we are giving away 1 aliquot, and in our agreement we're always asking, give us your results back. So then 4 other aliquots have, for example, sequencing data, then again proteomics data and other data, and this is like a good like cycle of science. So then I thought, like, is it biobanks only about samples? I asked AI, obviously, what are the key components in personalized medicine, and neither ChatGPT nor Google's Gemini said about biobanks. So I thought maybe it's not as necessary, but obviously we understand that biobanks is an important part of personalized medicine. So is it only about the samples and biomarker research? So there is tons of papers about biobanks in health, biobanks in personalized medicine, etc., etc., and they're all speaking about different things. But first of all, obviously, it's a standardized way to store samples, so to identify biomarkers, and then you can have large quantity of samples, so you can have a statistically good research. Then you can develop new cancer treatments, and for example, one of the cancer genome atlas is also called like a biobank, which can help to do that. Biobanks are also better cancer registries. It's because they can collect all the clinical data that are in cancer registries, but also they can collect the experimental data. As I mentioned, the cycle. You can get back more data than, you can get back data from clinical trials and from other biomedical research. And obviously collaboration, because usually fundamental scientists or industry does not speak on the same language as the physicians do. So biobankiers can be a translators, the bridge between fundamental science, physicians, and also industry. So collaboration is really important, and we understand that we need a sufficient number and higher quality and for the larger research projects. So there are more than 3, but the 3 that I would like to mention, collaboration consortiums is Biomedical in Europe, ISBER worldwide, and ESBB in Europe and Africa. And for example, if you as a scientist or biobankier, if you are as a biobankier, you should always put your information about your biobank and samples that you have to the Biomedical directory. And you as a researcher, you can always go there and search. There is a directory, you can search by country, for example, here, or by name. And already there are more than 500, less than 500 organizations, and you can search, for example, acute myeloid leukemia. Then you will find all the biobanks that have this kind of samples and data, and then you can open chat with them and start making research. So what's new with the regulations? So 2 new regulations, IVDR and FDA modernization. You know, IVDR 2 years ago appeared, and it says that if you want to sell your diagnostics, you need to validate them, verify them. So all the steps should be, you should prove the scientific validity, analytical performance, and clinical performance. And for all of that, you need samples and documentation. And for all of that, obviously, biobanks can help. That's why biobanks will become even more popular in few next years. And then again, less than 2 years ago, Biden signed the FDA Modernization Act that for the clinical trials, you can skip the animal models if you have enough proof with, for example, 3D models or like cell 2D models, organoids are coming of popular, or if you have enough sufficient data with your AI algorithms, then you can skip some animal models. This is again shows that data that biobanks have and samples that biobanks have could be used for the validation and to show that your drug or your test is working. So what are the new tendencies for biobanking? So first of all, multi-omics. So today, like everyone was speaking about multi-omics, it's not only about genomics. And I would say, like, we would love to make all the omics for one person, but you cannot take, like, for example, brain tumor and make all this all the studies yourself, because there is not enough sample, obviously. So there is a new tendency, I would say, to collect samples, for example, blood or skin biopsies, extract PBMCs, peripheral blood mononuclears, or fibroblasts. You perform reprogramming, the differentiation. You can have induced pluripotent stem cells, and then you can use those to have to differentiate them to different cell types, and then do different omics, for phenomics, metabolomics, proteomics, transcriptomics, epigenomics, genomics. And then from all that, you will get diagnostomics. So you will get like all the atlas about person. Then obviously personalized medicine, personalized therapies can be easily achieved. And you can use the proactive prevention. So for all that, new biobank collections, I would suggest that they should focus on viable cells. So you should definitely have more refrigerators that are minus 150 or liquid nitrogen storage tanks. A new tendency, again, it's not that new, and before speakers also told about it, it's circulating tumors, so free DNA. So it's less invasive, you know, can be repeated more times. You don't have to do biopsies or surgery to get samples. But it is not as sensitive, sadly, yet, but, and it can be affected by other factors, such as inflammation and other things. But, like, again, for biobankiers, my suggestion would be that collections should, your plasma collections should be of higher volume, because you need at least 5 milliliters usually, and we know sometimes more of plasma to detect those. The more you have blood, obviously, the higher chance that you will detect it. But most of the commercial kits to extract circulating tumor DNA need at least 4 or 5 milliliters. So you need at least 10 milliliters of blood to extract. Half of it will be plasma, and then you will be able to do it. And as a biobanker, you need more aliquots, so obviously you need even more blood for that. Ex vivo drug screening, as I mentioned, tumoroids, organoids. For that, you need, again, viable cells. And not just to store them in like liquid nitrogen, but you should extract primary cells. You cannot do it with all patients, obviously, as a biobanker, because it's too expensive, but you still can make a collection of viable cells, primary tumor cells or primary other cells, for the possibility to use them in ex vivo drug screening. And obviously, the future is also with the cell therapies, all different kinds of cell therapies. Personalized cancer vaccines are there, so I believe that in the future, we're thinking about more than 10 years in the future, biobanks should also focus on the GMP facilities, and because at the end we still want to do research that could be implemented and translated faster to the patients. So GMP facilities could do that. And for example, we in our biobank, in Lithuania, we have biobanks and tissue banks. So biobanks are for research and tissue banks for transplantation. So there are different regulations. So we collaborate, obviously, with them. And when they don't need some kind of cells, like stem cells or other cells, bone marrow, that they stored for a transplantation, then we can get it if we have consent from the patient. And then we have those samples prepared in GMP grade, and we can use them for scientific purposes. Yeah, short conclusion, with the new regulations, biobanks will become even more popular. We should focus on viable cell storage and prioritize lower temperature. And yes, thank you for your attention. If you would like scan, we have made our LinkedIn account of our biobank, small department, last week only, so we'll share some interesting stuff. And if you will have any questions or would like to collaborate, it's either Naumovas at Santa or Biobank at Santa.lt. Thank you for your attention.
Thank you very much for this nice presentation. So now we are heading to the last speaker, which is now an internal speaker, my co-chair here. Sorry, yeah. Monta, where is it? Yes, yeah. Monta Briviba. Sorry, I pronounce it right. So I just learned that it is freedom, that's a translation of your surname, which is very interesting. So we had already a good discussion during the break. So I'm really excited and look forward to your talk about your sequencing projects ongoing here in Latvia.
Yeah, thank you. So you just had a very good introduction in biobanking. It like messed my presentation, but okay. I will tell you about biobanking as a key scientific resource within, particularly within this Latvian Pediatric Cancer Initiative. And actually, who am I? I'm a researcher in Latvian Biomedical Research and Study Center, and I'm also maintaining childhood cancer biobanking within our national biobank. And I will start with the Latvian Pediatric Cancer Initiative, a brief introduction. It was established in 2019. It was actually encouraged by a patron himself, John Martin Tully, who managed to bring together Children's Hospital and Latvian Biomedical Research and Studies Center. So the project aim was very ambitious. It was to promote the development of prospective precision medicine approach in pediatric oncology in Latvia based on molecular analysis. And there were 3 objectives developed by that time. Firstly, to develop the pediatric cancer cohort in the National Biobank, then to perform this large-scale genetic testing to identify both the tumor-specific somatic variants and alterated gene expression profiles, and finally to create a multidisciplinary working group of both doctors and scientists that could interpret these genetic testing results and translate them into personalized treatment strategies, with the last one, of course, being the most challenging one. And I truly believe that all of this was possible due to already existing resources, and therefore I wanted to emphasize the role of the Genome Center back then. And the Genome Center has like 2 divisions. Firstly, we have this biobank, genome database of Latvian population, which maintains the biospecimens and also clinical data of more than 43,000 participants at this moment. And we are providing the necessary infrastructure for scientific research for international collaboration, ensuring sample processing, starting from the sample collection, then logistics, pre-processing, and also storage. We are also supporting biobanking of various sample types, from DNA to viable cells. We are ensuring linkage to corresponding omics data. Mostly those are genomic, transcriptomic, metagenomic data that are generated within BMC mostly. We are able to extract medical records from national registries. And finally, we are serving as a national node of BBMRI ERIC. The other part is the laboratory, where we are able to do both manual and bulk sample processing with different kind of automated platforms. I truly believe that we are the main next-generation sequencing service provider in Latvia. We are providing from small-scale applications on MySeq up to some high-throughput sequencing on DNB-seq T7 machine, which is here. And recently, we also acquired this great ProMathian next-generation sequencing platform, allowing us to also do long-read sequencing. And since we are generating a lot of data, we have a close collaboration with Bioinformatics Core Facility and also Riga Technical University High Performance Computing Center. Now getting back to the initiative. So there are 2 main collaborators here. Children's Hospital, of course, doing a great job in diagnostics and treatment, but within this project they are also doing patient recruitment, collecting clinical data, and of course tissue sampling. Meanwhile, the BMC is ensuring sample logistics, sample registration and storage in the biobank, NGS, and finally data analysis and data storage. And we have some interactions in the middle, some benchmarking activities, and we have had some pre-tumor boards together as well. So at the moment we have 427 patients involved. These are childhood cancer patients, and 166 of them are fully sequenced. And this is a schematic visualization of both the sample and also a data flow that was there in 2019. Anyway, we are still collecting such samples as a biopsy surgery material or bone marrow sample as a tumor representing material. The blood sample is used for plasma and serum aliquots as a standard procedure of our biobank. And we are also using blood samples, either blood or oral swabs, depending on the diagnosis, as the source of a normal DNA. Nevertheless, this schematic visualization has changed, and today it looks more like this. So it has become more complicated, mostly due to 2 facts. Firstly, We have much more interactions and collaborations with the Children's Hospital now, such as these pre-tumor boards that were not there before. Also benchmarking activities. We have several sub-projects going on, and now Biobank is also supporting Children's Hospital in some clinical protocols. And the second thing is that, of course, Children's Hospital has improved their capacity to do NGS analysis themselves, which is a great step forward, right? And now I will talk about this NGS approach in the BMC. So we are doing whole genome sequencing on this T7 machine. We are getting 70x coverage for tumor DNA and 30x coverage for normal DNA. And the RNA sequencing is done on smaller scale machine, this G400, where we are obtaining at least 200 million of 150 base pair-dense sequencing reads. The data analysis is done as a comparison between normal and tumor whole genome sequencing data pairs. We are identifying SNVs, indels, narrow structural variants, and copy number variants. The tumor transcriptome data are mainly used for fusion gene identification, and normal tissue whole genome sequencing data are also used for germline variant identification. And I still think that the main bottleneck is data analysis and interpretation, since we are gathering around 400 gigabytes of data per each patient. And therefore, I just wanted to emphasize that my dear colleague, Agnes Vilnius, who is a bioinformatician, she has spent last year by improving our data analysis pipeline. We are using some commercially available tools such as CLC Genomics Workbench and QCI Interpret 1, as well as open source tools such as the SAREC that I already saw in another presentation, and also, of course, some in-house solutions. And now we are finally able to interpret and detect and interpret the structural variants. We have also included microsatellite instability detection, and we are also doing gene expression analysis. Nevertheless, there is still a lot of things to do, especially in the field of interpretation, but it's also a matter of practice. And here's a very brief overview of the data. So here you can see the distribution of different diagnoses that we have in this sub-cohort that has been fully sequenced, and the majority of them are hemato-oncologic malignancies. Minorities are sarcomas, which corresponds to the global epidemiology of these diseases, so making the cohort representative. Then in the figure B, you can see a mutation, damaging mutation number per patient stratified by different diagnosis groups. I hope you can see the diagnosis groups. Nevertheless, the highest number of these damaging mutations can be found in sarcomas and hemato-oncologic malignancies, and these green bars are those damaging mutations, the median number per patient of those damaging mutations that are occurring in disease-related genes, and these are either drivers or some clinically relevant for prognosis, diagnosis, or treatment. And figure C actually shows you what are these genes. Unfortunately, you cannot read them, but what figure C tells you is that there is a huge heterogeneity between different diagnosis groups, and within each group, making each patient with his or her unique genetic background. So there's a huge heterogeneity within the cohort in general, and I counted only 50 genes affected, appearing at least in 2 cases in our cohort. Similar situation with gene fusions. These are clinically relevant ones, though we do have some gene fusions occurring repeatedly, such as this BRAF involving in central nervous system tumors, and also some related with Ewing sarcoma. So we are also able to detect these chromosomal aberrations. Most of them are found in sarcomas patients, so 93% of them. We are able to detect chromosomal amplifications, loss of heterozygosity, and this is copy neutral loss of heterozygosity. Regarding chromosomal amplifications, most of them are appearing in hemato-oncologic malignancies, and you can actually see a pattern there, meaning that they are mostly the same chromosomes that are involved. And an interesting thing that we also observed here is that there are no chromosomal amplifications in chromosome 16, no copy number loss of copy neutral loss of heterozygosity in chromosome 16, but we do have a loss of heterozygosity in general in the same chromosome. So that's an interesting fact. And now I would like to get back to the biobanking. I would say that the most effort at the moment is for supporting the contribution of hospital in clinical protocols. And here we are, as a biobank, we are ensuring sample pre-processing and storage. We are already participants of Umbrella protocol. We have developed our internal laboratory protocol for all together, and now looking forward to also participate in CHIP AML. This is just an example of one part of the laboratory protocol, just show you that there is a sample stratification, and what I see challenging in this is that given the small volume of these samples, it is really challenging to plan how these samples will be stratified within one protocols, and even more challenging between different protocols. And then there was one sub-project going on, longitudinal gut and oral microbiome study, where we are longitudinally from the same patients collecting both stool samples and oral swabs during the therapy. We are running shotgun metagenomics for stool samples and 16S ribosomal RNA sequencing for oral microbiome analysis. The clinicians have done a tremendous job in developing these extensive questionnaires that are involving food frequency questionnaire, lifestyle and clinical information. And we are doing this microbiome compositional analysis, and also planning to do the antimicrobial resistance analysis, and hopefully also some metabolome. And recently this protocol was revised, and we are also looking forward to involve healthy controls. For those who have worked in that field, you know, healthy controls are pretty challenging if we're talking about children. And we are looking forward to collect stool samples from healthy siblings of the same patient. So I'm very eager to see what the data will show. Oh, sorry. Yeah, and I really believe that collaboration, given the small population of these childhood cancers and this huge heterogeneity, I truly believe that collaboration is a key step to ensure relevant scientific findings. And we do have some very great collaborations. We are hoping to become Childhood Cancer and Leukemia International Consortium partners. We are involved in this Can Heal project. We have a great collaboration with Oslo University, with Lars. We have a collaboration with St. Jude Children's Research Hospital, where during upcoming months there was one exchange visit planned, and we also recently became a part of the Yunkan Consortium. And there are many people involved, of course, most of them are representing Children's University Hospital of Latvia, but here I just emphasize mostly the ones that are from BMC. Thank you, everyone. And while I still have your attention, I would like to kindly ask Latvians, to participate in the Latvian Population Genome Reference Project by scanning this QR code, filling in the questionnaire, and later on donating a blood sample. So thank you very much. So maybe I could invite all of the speakers to take a seat here. I think we can start a question and answer session, right?
Yes. Maybe Monta, you had one question to Johanna already.
Yeah, what was it?
Yeah, okay. Is there?
No, I did have, regarding those scientific projects that you mentioned, you had this great table, many projects going on. Are these scientists returning data back to you, or no?
Is it on? Samples, and they do some kind of analysis that they should return the data. But of course, that has to also be in their ethical requirement, and we have to have some data agreements. There is a process for that, but that is the policy, so we can have the data back, and of course share it to the third project or fourth project later on. So that's the aim here.
Oh, it's great. If I can continue, I have actually a question to you. You mentioned that for the majority of cases, you also have this primary tumor available tissue, right? And then, I mean, primary tumor tissue together with cerebrospinal fluid. But what about the cases where you cannot access the primary tumor or it's too small to take the tissue for your sampling? Are you still focusing only on the findings that can be found in cerebrospinal fluid? Or how are you doing those?
No, so we get, from most patients, we get tumor tissue and do whole genome sequencing. And then when we can't do that, then we use the FFPE blocks to extract DNA and then do exome sequencing on
okay,
these.
so you have these FFPE.
Yeah, there's one question.
Hi, I'm not sure if it's working, but you hear me anyway. Marie from St. Anna Children's Cancer Research Institute in Vienna. I have a question regarding the consent. Since we are working with children's samples, do you reconsent when they get older? Do you reconsent when they're 18? Do you have opt-out in your consent? How do you deal with that?
Yeah, we reconsent when they turn 18, and they have the possibility to say no to further use of their samples. Mm-hmm. And that's also a huge workload for which you need a research nurse to coordinate. And it's difficult to get funding for these research nurses who are there to do the things that are not that visible for their funds and in the research papers. So.
Yeah, for us in Sweden, actually we are not legally obliged. We don't need to reconsent if the parents have approved this biobanking, but we have decided that we will do it anyway from a more ethical point of view. So we have not started yet, but we will start with this procedure, and it will be when these are turning 18 and coming back to the clinic anyway for a last checkup, going to the adult healthcare system, so we will have a routine for that. So, and then there will be opt-out possibility then.
Yeah, exactly the same for Lithuania. So it's not obliged. We don't need to do that, but we are creating a strategy how we will reconsent it. And usually all the consents have the information that will recontact person. So in the first consent we have this information that we can recontact you, and once the person will get of age of like adult, then we will be recontacting them. But it's like not according to the law, but only according to the ethics.
Yeah, well, very similar in our country. We don't need to do that according to law, but maybe something will change when we have this new biobanking law in place, right? And we also do have the necessary information. The patients have agreed, these particular patients have agreed to be contacted again, so it is doable as soon as it is necessary.
If I may add to that, because we had this discussion already yesterday when we had a short meeting. So Norway is the same, that we have to ask when they turn 18, so we need new permissions. And so we discussed that yesterday, and we said that it would be nice to have an app. So where you, I mean, in some countries you have already the app where all your health data is registered and where you can connect. So if you would get then an informed consent by that app, so that you could agree or neglect to get it back. But it's not implemented yet, but that's something all of us would like to have in the future.
But it's interesting to know that you only reconsent when they turn 18 and not with the age jumps you have before, because at least for us we have different consents depending on how old the child is, from 10 to 15 and so on. But you would not reconsent in every age group you fall into, but just with 18. That's good, because otherwise it's a lot of work.
Yeah, and it's also different. In some countries you need the permission from both parents, and in some countries it's sufficient if you have it from one. Are there more questions from the audience? I would like to ask Torben two things. One thing first, it's wonderful what you said about the study nurses. We also have a very dedicated study nurse, and so we are really dependent on these kind of people who are so dedicated. So I really appreciate that and can just support that. I would like to ask you about the cell-free DNA. And I mean, you have the Tumour cell-free DNA and the general cell-free DNA. So we know that stress reaction of the body can create some cell-free DNA, and then there is some which is particular from the tumor. Do you think we could separate these two cell-free DNAs, or see if which one really comes from the tumor and which is just a stress reaction from the body?
Yeah, it's difficult to do. You can get some indirect analysis where you look at the length of the cell-free DNA, because the tumor DNA is a bit shorter than the cell-free DNA from the healthy cells. And then you can do this digital droplet PCR where you look at the tumor-specific mutations also and get an idea of how clean your sample is. But it's a very interesting area also where it would be interesting to sample a patient over time also before you do surgery and see if it changes during the day and if factors as fever will influence the concentration. Yeah.
Another question I would like to add is that the vesicles, because we know that some of these vesicles, they contain cell-free DNA, but I mean, you have small volume sizes, and then when you extract the vesicles and then extract the cell-free DNA, it's getting less and less material. Do you have a good method to proceed with these small volumes?
Yeah, we haven't extracted the DNA from the extracellular vesicles yet. This study which I showed was in collaboration with a scientist who recently came from Lund University, and they have focused on the metabolome and the syndican one. But I really think it's an interesting biomarker as well.
Last question. MicroRNA, you stressed out the importance of that. I'm interested in long non-coding RNAs. What do you think, are both of them equally important, or do you think that microRNA are in a way the main trigger or cardinal trigger for some, or could be used as a marker better than long non-coding RNAs?
Yeah, so it's really interesting with the non-coding RNAs, and it's an interesting area also because it's sort of an isolated area where some people, they think, well, it's non-coding, so it's not really interesting as compared to the coding RNA. We're collaborating with a researcher at our university in Aarhus, and he's looking at the long non-coding RNAs and has a model where it looks like in a single cell sequencing project that some of the tumor cells which looks like they're about to become metastases, they have a different RNA profile of the non-coding RNA. So I think microRNA and long-coding RNA have different functions, and that the future will show us that it's important to look at as well.
All of you stressed out the collaboration and the importance to share material and data. How difficult is it? I mean, across borders, across different legalization and different rules, and so I would like to hear of all of you your experience, how to share both material, data, collaboration. When it's one thing is inside Europe, but as well if it comes then to America or other places, how difficult is it? Which experience do you have, and which advices could you give to us in here?
You mentioned that you had shared some data or material.
Yes, so for all these projects that I mentioned, it's mostly in Sweden, so that makes it a bit easier, especially for biobank samples, because according to the Swedish biobank law, to be able to share samples from Swedes, you have to have a Swedish ethical permit. So we cannot send samples abroad with an ethical permit in another country, but the Swede can have a collaboration and a Swedish ethical permit and send then samples for analysis with the collaborator in another country. So that's possible, and that happens a lot. You write the MTA, you can do that. Data you can share directly. We have just one, as I mentioned, this example with Heidelberg, where they have requested genomic data from us, and that is there is processes to approve this, and especially in the European Union with the GDPR, it's possible, but it takes some time always with the regulatory and with the data transfer agreements and make sure there's ethical permit. But we have routines for this. But for the United States, we have had some requests, but we have not sent yet because there seems to be a legal issue there where lawyers say most of them are reluctant to for us to share actually, which of course is a great disadvantage for the whole research community. So, but it's a problem.
So the data that you shared with Heidelberg, do they keep the data, the genomic data, or will you give it to them and they do the analysis together with the rest of the cohort and then return the genomic data?
Yeah, actually for that study, actually it's not, we have not actually signed the last DTA yet, so we have not sent data yet, but we have approved their request. And actually from the beginning, this is a clinical study where they from the beginning wanted to collect samples, but they were going to send the samples to Heidelberg and do genomic analysis. And we said, but we are doing genomic analysis on the exact same samples already in Sweden, so it's better if you get the data, we don't have to double analyze the sample. So then in the Swedish permit, then they also tell this to the patients that consent for the study, that the data will be sequenced in this project, but you will share it to the Logic project and send it to Heidelberg. So it's very clear. So sorry, what the new question was? Sorry.
That was if you give the genomic data to Heidelberg and then they keep it there as part of their...
Yeah, exactly. And then they are approved to use it in that study. So they will keep it, of course, for the purpose they need it for that study. And that's how we approve it, that you are supposed to use it for what you have wrote in your ethical permit and application to us, and not maybe share it further. But if they want to share it further, they can maybe make a request for that as well later on. So, but
so the patients they were informed and gave written consent to it. Is that so? Is that necessary to get data from your biobank, or if we have a project where we want to look at MYC-amplified medulloblastomas, can we then?
It's different, and this is up to the ethical review board actually to decide. So for some projects, they don't have a specific consent for their project to the patient, but they have very clear written in the ethical permit that we would like to use the data from the Swedish childhood tumor biobank. We do not intend to collect the consent for our study because this child or parents are already consented to the biobank project, where they also, in our consent, is very clear that this data can be sequenced, we can do molecular analysis, the samples can be analyzed, and it can also be used in other projects that are ethically approved. So already they know from the beginning that this can be shared further on to ethically approved projects. But it's up to the ethical review board to say, okay, this is a very specific project. You need to contact the family and say that you will use the data in this project. So,
Daniel, you have made the same experience?
Yes, I would say for Lithuania we have a bit easier than in Sweden because we can share it across Europe and also other countries. But in order, we can have two-way collaboration. So the same as in Sweden, we can have a collaboration, then we get a permit in Lithuania, and by ethics, ethics committee will approve for a study in Lithuania, and we then can share according to this permit. Or if they have approval of their own, in their own country, from their own ethics approval, then we need just a copy of approval, and then I am, as a biobanker, I need to contact ethics committee, Lithuanian ethics committee, and tell them, look, there is this approval from their ethics committee, they will do this short summary, and I want to share this and that data. So there is two ways, but usually, obviously, it takes some time. It's around 1 month to get approval from our ethics committee, and usually takes longer to get agreement between countries. And it's much easier with European Union countries. We have shared with Italy, Ireland, Germany, Sweden, without any bigger problems. When we are sharing with US scientists, we do that, but we usually ask them to give approval that they will stick to the GDPR rules. So it's easier to share samples than data, but usually you never share just samples. You always give also some data with it.
Yeah, about our situation, it's something similar. Each study that would like to get the data, either data or samples from the biobank, must get firstly the ethics approval, and also we have this Genome Research Council. So there are 2 institutions reviewing this project. And when those approvals both are in place, it is possible to share both the samples and the data. Within Europe, it is happening pretty easy. We have MTAs, DTAs, depending on the occasion. I actually have a recent experience with sharing data to St. Jude. And again, you must stick to GDPR rules. I got the ethical approvals, and then we came to this DTA, which foresees the GDPR rules. And St. Jude themselves told that they are not able to fulfill these criteria that are fixed in GDPR rules. So unfortunately, we didn't manage to do it eventually. But St. Jude actually is working on it. They are improving their infrastructure and standard procedures to fulfill these criteria.
Thank you very much. If there are, that's yeah.
Just again, a more practical question. How do you manage to get all those fresh, frozen samples and like share with pathology? What was your strategy there? Because I mean, if you have at time point of diagnosis, you have only like 2 small fine needle biopsies, and so for us, it's always a bit of a fight with the pathologists, obviously, because they want to keep everything because they need to make the final diagnosis on it as well, which is a fair point. But then, how do you get samples for the biobank from cases like this, or how is this organized?
I think it's all begins from the infrastructure and how you have like agreements internally in your hospital. First of all, in our hospital we have like a vacuum post that all the samples are going not by hand, like nobody is doing it, they are flying like for the basement. So because our pathologists are, for example, 300 meters from the main hospital, so first of all the samples fly fast. So this is faster than just walking and bringing those samples, and then you need a good IT solution, I would say, that the surgery person would put the timestamp of the ischemia time, and from that you will calculate the time. And then usually if it's an important project or important collection for biobanking, there will be a pathologist will be sitting on the phone and waiting for the sample, and he will do as fast as he can, split the sample and share it with, and freeze it. For example, if they have a liquid nitrogen like infrastructure, and then we take samples, for example, every week or every month if they have infrastructure. If they don't have, then samples fly back to us by the vacuum post, and we store them at our biobank.
So it will be for sure always fresh frozen as well, and not everything in formalin.
They are storing in formalin like also for their like diagnostics, and we will get that also to the biobanking later, because we can also get diagnostic samples.
But from all tumors, you're able to save material for the biobanking.
From the same material, yes. So
you always get the biobank, because I understand your question, because we don't get. That's why I'm showing my figure from. Fraction of the patient, I don't know, maybe 20-30%, we don't get the tumor material because it's needed for the diagnosis, especially when you have the small needle, fine needle biopsies. So that is, of course, a problem, because diagnostic always go first, and some childhood tumors are very small. But I think it's also the communication with the surgeons and pathologists, of course, to really try to save for biobanking, because it's not only us as a biobank that want the sample. It can be also, of course, other clinical studies, other research studies that wants the samples from the same patient. So that's it's an issue.
Yeah, obviously, sometimes you don't get the sample for research for biobank. First is diagnostics, and sometimes you have collection only with blood or like cerebrospinal without tissue. But obviously, the ultimate, and it depends on the cancer, how much the surgeon removed this cancer. You know, how much you can use it for the future research.
Yeah, I want to be honest. That is also a fight, but we give them we give the pathologists a lot of love, and then invite them into the projects, and then by time it seems like the collaboration becomes better and better. And then we've been a bit naughty also, because with the pediatric brain tumors, we actually just ship the samples directly from surgery to the Genomic Institute.
We're not allowed to do that anymore.
No, it's also secret. But it's sometimes the pathologists say they never get enough tumor tissue, and they're also a bit offended by the nanopore sequencing, for example, that we can get diagnosed before they even get the samples in the hand. So, but I think the trick is to involve them in the research, and yeah, we won't move forward if we keep fighting about the samples, right? So.
But we struggled in the beginning to have it fresh frozen, because we started more than 14 years ago, and then it was in formalin. But then when you set the routine and talk to every new nurse that starts, and to get the routine in place. So now I think it works very well to get them fresh frozen if we can get material.
Yes. Then I would like to thank all the 4 speakers. It was a wonderful event, and thank you as well for the questions from the audience. So maybe we give them another hand.